talk today is going to be about getting outside your comfort zone. It's important to always do things that you may not want to do or you may not feel comfortable doing, because this is the opportunities in which you'll have a great chance to learn something new. So that's what's going to be the theme throughout this talk, and I'll do my best not to bore you with the strenuous research details. We're just going to be hitting the highlights from my 2 weeks there. So this is Dr. Stephen Badlock. He is an incredible man who is so intelligent. He is, well, technically he was my boss while I was there. I worked under his lab, the Badlock Research Lab. They do a lot of work in regenerative medicine, extracellular scaffolding, and things like that. As you can see, he's very accomplished. But my favorite thing about him is his passion. This man loves to think outside the box, which I think is so great and something that we should all try and do every day. So he's a vet, but he also works in research, which I think is great because that shows that people like us, if we chose not to go into general practice, could do research in the future and help humans and animals as well. So here's my one and only picture of the lab, because my contract when I went there, you're not allowed to take any photos inside the lab, so there is the lab and all its magic. I have a few photos from some histological slides and things like that Dr. Badlock approved, saying that I could use in my presentation, but as for the lab itself, this is kind of what you guys get. But imagine mad scientist lab, bubbling things everywhere, and that's kind of what it's like. So we'll go ahead and get started. So what was I doing while I was there? I'm really big into neurosciences, but I'm also a vet student. So they tried to incorporate me into things and projects that I would enjoy. So my mentor, Mark Murdoch, he works in glioma cells. So my research while I was there, we worked on these human glioma cells, looking at exosomes. I'll get to what an exosome is. I had no clue what that was until I arrived there. So we worked on the glioma trials, and then we cultured them and did some PCR. You got it? Yeah. And then the hydrogel therapy, that's going to be the last little bit of the topic, and that was the part I enjoyed most. We got to work with rabbits, so it was time for me to shine because I actually know things about rabbits as opposed to the rest of the research we were working on. So we'll go ahead and get started. So what is an exosome? These essentially, in a very layman's terms, are little tiny vesicles that are all over the body. And what we were doing with our research is you can actually specialize the cargo inside the exosomes to perform a certain task. So we were looking at adjusting the surface markers on the exosomes, so they would specifically target the glioma cells, and we could use this as therapy in adjunct to chemo and things like that for tumors. So here's this awesome diagram that my mentor Mark used to explain what an exosome is and how we get those from the cells. So we'll start over here at number 1. Wow, look at how shaky that is. So essentially what happens here, I'm not going to point from now on. Anyways, number 1. So what's happening here is that inside a larger vesicle that exosomes are formed, and they're essentially just tiny little vesicles, so it's a bunch of bubbles inside a big bubble, is what we're going to describe it as. And what happens is once those are produced and they're mature enough, that larger vesicle gets sent to the surface of the cell, and those exosomes are released out. And the way they work is to have surface markers, which kind of tells them where to go, so it's all cells in the body. So when they find their cell, you can see over here, there, number 4 and 5, when they find their target cell, they bind and they unload their cargo, which performs whatever function is needed at the time. Over here in number 3, you can see that as all things in the body, we have our own defense system. So if the exosome that was produced isn't 100% up to the body standards, then it gets fused with a lysosome, and then that degrades the products, and then it's basically a non-functional exosome. Okay, there's your quick background on exosomes. So these are my awesome sketches from my notebook while I was there. There's going to be a whole bunch of those in the presentation, so just bear with me if you can't read my handwriting. But essentially what we were doing is we have 3 cell lines of human glioma tumors that are labeled there. The numbers just correlate to the dates in which we received those specimens. So from our master sets, we decided we were going to culture a cell line from each one of the master mixes. And that's what you can see here. So it was a 6-well plate. Each cell line got their own plate. That's how self-culturing goes. Sorry guys, I'm trying not to bore you with the details of it, but that's what it looks like. We used collagen and then PBS, which is just phosphate buffer solution. It's just a medium that you can use to grow cells everyday kind of stuff. Okay, so when we got down to using the numbers of determining how many cells we wanted to produce in each one of our wells, we had to go back to the literature. In research, you do a ton of reading. It's an insane amount of articles that I had to like look at and like try and decipher through to figure out how many cells per well we were going to use. So after like, I don't know, like 30 or 40 articles, I think, and like 2 lunch breaks later, we finally determined that we were going to use about 500,000 cells per wells. And then for the exosomes, on average, most people use about 25 microliters for their exosome treatments. So that was kind of just like the general that we used, going based off the literature. So as we proceeded forward, oh, just kidding. Let me talk about what our exosomes are supposed to do. So we had altered these exosomes to basically inhibit mRNA. And their cargo inside contained a microRNA, which essentially chews up the mRNA, enabling it to do its function. So we're halting the cell function as a whole using the exosomes. So here are my glorious glioma cells. Or super hard to culture these. This is like my second attempt because my first attempt didn't go so well. So all we can see here is our different cell lines. So this top one is going to be 0319, and the bottom one is 1015. The only thing that I'd like you guys to notice is that the cell morphology is a little different. That makes sense considering they came from 2 very different people and they were 2 different tumor types. The top row, that's considered a high-grade glioma, and the bottom one is considered a low-grade glioma. Anyways, I thought they were super beautiful, and I really like using this fancy microscope that has the camera. So here, the next step, we were doing the hemocytometer, and that is, wow, that is like a really challenging thing to do. Essentially, it's like a little glass slide, and it has a grid that you can see in my fancy drawing up there. And then what happens is you take the cells from your culture, and you dye them with tryptophan blue, and that dyes the dead cells blue, essentially, and then you just count them. And it is tedious and time-consuming, and you have to be consistent and patient, because there's a certain rule of thumb that you only count like 2 sides of each of the square. So if there's cells that fall on all 4 borders, you have to pick 2 of the sides and you count those, and you have to be consistent throughout. This is what Mark had me doing for like an entire day, and it is exhausting to just count dead cells. Also, it's not very rewarding. So then we continued on forward with our project, and our next goal was to extract the RNA that was left and see if we had any viable RNA molecules used for the rest of the project. So when it comes down to extracting the RNA, this is yet again another tedious process. Who knew that research could be so much work? And then when we finally got them all mixed, my sweet drawing is an excellent representation of what it looked like. So the RNA is at the surface, and then there's a protein interface here, and then the bottom part is DNA. And they're divided out based on their molecular weight. So RNA obviously weighs the least amount, and that's why it's at the top of the tube. So when you're extracting the RNA, you have to use your pipette and a very gentle hand to only suck off the RNA interface and not get the protein. If you're like me, you have really shaky hands and struggle to do this. So Mark ended up having to do the vast majority of it because we wanted to save the experiment. So once we've gotten the RNA removed from our tubes, and we had that for the next step of our project, we decided to do an nanodrop. So it's using a spectrophotometer. Basically, it's like mass spec. So we're just counting the number of RNA molecules per 1 microliter of our solution to ensure that there was enough RNA to proceed with our electrophoresis to make sure that it had the appropriate cell markers for our exosome treatment. So you need about 1,000 molecules per microliter. Ours obviously were like above and beyond that, which was like super exciting news. This again though, like everything else in research, is really tedious. 1 microliter is nothing. And this little tiny, it's like a conductor. You basically put the drop on, and then the machine like sends current through it and counts the molecules. So I got to do that, which was, it's exciting stuff. And then electrophoresis, everyone remembers this from like high school biology, right? The positive negative current pulling molecules across. Yeah, everyone looks like they really enjoyed that back in high school, huh? So that's what I got to do. Here's my awesome representation. We ended up using a ladder, which is what the L represents. It's just going to be our control to let us know the number of nucleotides and how far they stretch, so we have something to correlate our specimens and samples to. Here is my awesome table Mark and I developed. When you do electrophoresis, the gels all look exactly the same no matter what you put in them. So you have to have these tables to help you decipher what it is that you like put in each well and which samples you're looking at, what genes you're going to be studying. So I have a whole bunch of these. And here are my glorious little gels. We took these photos using a really cool machine that I don't really know how to use because it was like radioactive, and Mark told me it was too fancy for me to touch. So he ended up taking our pictures. And this is our ladder here. This is the way it's supposed to look. It's just overexposed. This is a bit more of a normal ladder. And then we're looking at these faint lines. I know they're gonna be really hard for you guys to see, but they're there. And then these obviously are a little bit more brighter. And these are just letting us know that the certain genes in which we were like studying presented on our RNA molecule. So it was like a green light that our exosome treatment could work on this particular cell culture section. There's gonna be a whole bunch of these, so there's like 3 or 4 more. Here's another one. It's not as bright, so it's a bit more of a, not like a 10 out of 10, maybe like a 6 or 7 out of 10. So the therapy could work, but it wouldn't be ideal for that particular patient. And then our last table, this is like a nice one you can see here, all across the board, meaning that's a 10 out of 10, so that particular gene was present throughout all the RNA samples that we collected for that particular cell line, so the exosome treatment would be most ideal for this patient. Lastly, the hydrogel. This is a really awesome product that unfortunately didn't work the way it was supposed to. So what it's designed to do, what the pharmaceutical company had hoped, is essentially when they open up body cavities and do surgeries on patients, this is particularly for humans, but we use rabbits as our model. They, let's just say that you made an incision, a ventral midline, and then you just like took some sandpaper and roughed up the uterine body, which is what they did in the study here. The whole purpose was that this gel was going to be squirted onto that, and then it would inhibit adhesion formation, which seems really cool. And I thought, wow, like wouldn't this be fascinating for like the real life? And then, sadly enough, in our pictures that we'll get to, adhesions were formed everywhere. It was like a total bust, unfortunately, which I guess happens a lot in research. It's not always going to be a 100% chance that things will go right. So here's our fancy table that we came up with to grade the adhesions. We tried to be very subjective about it. So Mark and I worked with the rest of the research team there at the Bad Lab on coming up with a table that would be easy for like people to follow if we pulled someone off the street and asked them to grade this particular adhesion. So here's our rabbits. They were the cutest little rabbits ever. And then here they are on necropsy. The photos they're better on my computer, sorry guys. So here is a nice juicy adhesion. Unfortunately, that's not what the gel was designed to do. This is a bit more normal, so this is just some fibrosis here on the abdominal wall. Not a big deal. The gel kind of sort of helped here, but wasn't ideal. And then in the next couple slides, you can see here's another adhesion. And then the ascites, and then there's fibrosis here on this wall as well. So unfortunately, the hydrogel project was kind of a bust, and there was insufficient data for us to continue further with human trials. But the point is that at this moment in my time there, I was no longer a novice. Like, I knew more about rabbit anatomy than my mentor, George and Mark. So it was time for the tables to be turned. They got to be in their uncomfortable zone, and I got to be back in my comfort zone. That's what I want you guys to be always aware of. Even though like you're in a place that you don't feel like you fit in, or you're like not quite there, like just remember, there's always a moment when you can like teach someone something new or help them through a project that they might be working on. And that's what I got to do here with the rabbits. Even though I don't know a lot about rabbits, I watched like 6 YouTube videos on like rabbit necropsies to make sure I knew what I was talking about. It was just really cool for me to be able to share what I've learned with people who know absolutely nothing about rabbits. So that, I think, is the end. Here's this awesome skyline photo of Pittsburgh that I got to see every day when I walked home. And I want to give a special thanks to Dr. Badlack and my mentor Mark, and then to Dr. Gravemeyer for recommending me for this externship. So yeah, thanks for listening to me and eating pizza.